Okay, so I hope the coffee has not erased your memory of frustrated by stability on these oscillations. So what I want to do now is to really bring all this back to yeast. So let's talk about what happens in Sunil's chemostat. Maybe I don't need so much of the board, so I'll stay here. So let's try and list the kind of processes. So I'm going to still remain at a somewhat abstract level that I took from Sunil. So remember, I had taken sort of the fact that there were two states, Q and G. The fact that there were, the total number was not changing, but there were oscillations between Q and G. So the numbers of Q and G were switching back and forth. And there were, as Sunil is drawing now, there were two time scales. So how can we take the ideas that we were talking about in the previous lecture and do exactly address the question that was raised? How do we think about this now at a population level? So we are going to make a leap. We are not now, I'm not going to talk about molecular stuff happening inside a cell. I'm going to talk about a population of cells. And some of those cells will be in a Q state and some will be in a G state, and I will take that as given, that there are no other states. So let's think about the kind of processes that can happen. Sunil mentioned that you can get switching from Q to G. So let me call the switching rate be QG, switching rate the other way be GQ. If that gets confusing, tell me, we'll change the notation. But I hope you can keep track of it. GQ means G to Q. So switching can happen. Other than that, one of the states is proliferating. So we have to account for the fact that a G cell could give you two G cells, and that will happen at some rate, gamma. We'll come back to what these rates would be. Would they be constants? Would they be changing? What would they depend on? But for now, I'm just going to call this some kind of growth rate. Other than this, remember, it's a chemostat. So there is also a dilution. There is an outflux. You are putting stuff into the chemostat, typically nutrients, oxygen, maybe some other stuff. I don't know what all Sunil needs to keep these things alive. But he needs to put in various kinds of stuff to keep the whole thing alive. That means he's adding a certain amount of volume. So he needs to be taking out volume to at least keep the volume constant. But as you take out stuff, you will also take out cells. So your Q cells will die. Let me call that some dilution rate delta. So they will go to nothing. And the G cells will also get diluted at the same rate. Right? So this is switching and growth and dilution. So let's try and write down what would be, so if Q is, I'm going to, tell me if this is confusing, but if it's okay with you, I will use the letter Q to represent both the state as well as the number of cells in that state. Is that okay? Just for a bit. I hope it won't confuse you because I will, so I'm going to write down this kind of an equation is going to tell me something about how the number of cells in the Q state will change with time. So of course, we have to look at all these processes, and you will get a minus term for things that switch away. I'm going to use a linear term. This is just more or less a definition of what this rate means. So every Q cell has a certain probability per unit time to switch and become a G cell. And later on, we'll see that I might make these probabilities per unit time depend. These rates depend on other quantities. Maybe they depend on Q itself. Maybe they depend on how many cells there are. But for now, let's not get into those complications. I'm going to write a switching rate away from Q, a switching rate from G to Q, right? And a dilution. So the dilution is going to take it away. I'll write a similar equation for the growing cells, number of proliferating or growing cells. And they will have very similar kind of equation, but there is a growth term, which is not there in the Q cells. So I need to put in something like this. There is a, so I'm assuming exponential growth. We can come back to whether that's a sensible assumption or not. That's something that Sunil may be able to answer you, but for simplicity, I'm just going to take exponential growth. Then we will get the switching terms, except they'll be the other way around. So you will have minus BGQ G minus BQ G Q, sorry, plus, and then a dilution term. So far, so good? If anybody has any problem, stop me at any time. I'm just translating these sort of chemical, like chemical reactions into a chemical rate equation. Except that these are not chemicals. These are numbers of cells. So for those of you who know how to write down chemical rate equations, it should be very straightforward for you to go from here to there. But if you're not, don't worry, just think about switching, growth, and dilution. And these terms just follow. Yes, I have included that here. So the same dilution, no, that won't come in here, right? Because the number of the dilution process, I mean, there is dilution of the Q cells and dilution of the G cells. So this dilution, I'm imagining it as the following. A certain volume, a random volume of this chemostat is just removed in order to make sure that the volume doesn't change. And what cells end up in there are random. If there are lots and lots of G cells, you are likely to take more G cells out. If there's lots and lots of Q cells, you are likely to take more Q cells out. If the volume you're removing from the chemostat is just random, this is a well-mixed system, remember? So the chemostat volume is all mixed up. There's something actually stirring it, I think physically. So what you remove, which molecules you remove are random. So again, the dilution acts and removes things in proportion to how many there are. If you double the number of growing cells in the volume at a given time, then in a small unit of time, the amount you remove in dilution will double. Fair enough? These are assumptions of various sorts. Yeah.
In growth equation, you wrote G is giving to 2G. So while writing equation, are we not considering the coefficient?
Well, you know, so
like
you only get one new G effectively. It's like you are creating only one new G. The 2 also counts the one you started with. So here, I mean, you're asking why did I not put 2 here, right?
Usually when we write a differential equation out of reaction, so whatever coefficients it is there, it comes in concentration of that molecule or whatever you are taking to the power square.
So you
G
won't
square.
get a, so you're asking why not a G squared?
Yeah, yeah.
You won't get a G squared, you would get a G squared if you needed 2G here. If you start with a G, so if you start with a G on the left-hand side, and so think of it as a chemical reaction. If you had A going to B, then dA by dt is simply minus some K A. If you had 2A, then I agree, you would have K squared. In fact, you would have minus 2 over here. But in this case, firstly, you only have a G, so you only have something like this. Secondly, over here, you're coming back and you're producing 2A. But the net production is one new one. So in the net, you would have one new A. Okay? Any other questions? Because everything is going to be based on these equations. All I've tried to do is put in these processes. So let's just continue and write down the total, because I actually want to make sure the total is constant. If you look at the total, lots of things cancel out, and you only get this. So if you remember, Sunil said that the total is constant. You can only keep the total density constant if your dilution is not a constant over time. This kind of makes sense, right? Intuitively also, you have oscillations, There are periods of time where nothing is dividing, nothing is growing and dividing. And there are periods where there's a lot of cell division and growth happening. If you had a constant dilution, you would also see the numbers go up and down. There's no way you would not see the numbers go up and down. So the chemostat is working in a mode where there is a feedback to the dilution rate, which keeps it constant. And we have to take that into account. So the density of the cells is constant. And in essence, the number of cells I'm going to assume is constant. In order to do that, this quantity obviously has to be something that is proportional to the growth and the number of growing cells. Okay? So what I'm going to do with all this is transform this entire equation and describe the whole system not by, two variable, number of Q cells, number of G cells. Since the total is constant, I only need one variable to describe it. And I'm going to choose the fraction of Q cells. And it's very easy from this to get the fraction of G cells. That'll just be 1 minus. Okay? So I'm going to write down an equation for this, and then I can forget about all that. I don't need multiple equations. One equation will do. And then we can use all our tricks that we talked about with one dimension to get bistability, to understand how do you get bistability. So how do I get this from here? I just divide across by G plus Q. I mean, we can do this more rigorously, but I don't want to spend too much time on deriving these equations, just to give you a sense of how it's done. But roughly speaking, divide it all across by Q plus G. So you will get dQ by dt, d small q by dt over here. Okay? You will get minus bqgq plus bgq 1 minus q minus gamma 1 minus q. So I am going to use this equation a fair amount. So there is, essentially this equation is the same as that equation. I've not done anything very profound here. I'm just saying the total number of cells is constant, so let's just talk about the fraction instead. And using all this, I've just got that. And they correspond exactly to the same things. This is switching from Q to G, this is switching from G to Q, and this is dilution. And dilution now starts to look a little nonlinear, but that's simply because the dilution rate has to vary with time in order to keep the total constant. Any problems with this equation? Let me know. Okay? All right. Before you question me too much, I will get rid of the other equations. So what I want to do now is to ask, how do you get bistability from that at a population level? So it comes back to the question you raised at the end. Can you get bistability from this? So let's use the techniques we've seen in previous lectures. Suppose VQG, VGQ, and gamma are all constants. Let's assume that's the simplest case. Let's take that case. So I'm going to draw Q. We know Q goes from 0 to 1, right? It's the fraction of cells, so we only need to worry about 0 to 1. Let's draw some of these lines. This term, all the switching combined, you can see is a linear function. If BGQ and BQG are constants, this is linear, right? It's a straight line. When Q is 0, the only thing that will survive is BGQ. So it's a line that starts from BGQ at Q equals 0, and when Q is 1, you will have minus VQG. This term will go to 0. I'm only drawing the first two terms. So somewhere here, there is minus VGQ, and you will have a straight line joining them. What about the final term? The final term, gamma Q1 minus Q, is going to be something that looks like this. This is going to be gamma q 1 minus q. Does that make sense to people? When q is 0 or q is 1, this is 0. So it's fixed there and there. And it is non-linear in this fashion. It goes down and it comes back up. So when q is small, this is minus gamma q 1 minus q. Yeah. Minus v, sorry. So I should be careful. Yeah, this is qg. Thank you. Thank you for pointing out. That's important because once I start varying parameters, we'll need to keep track of all this. Okay, right? How deep does this go? That depends on gamma. If gamma was very small, then it would be just a curve like that, right? Not very deep. So if I add all this up, what's going to happen? So now I'm adding it up and I'm drawing all of that. Essentially, if gamma is very large, you might get something like this. If gamma is kind of smaller, you might get something like that. Well, maybe I've drawn it really badly. Let's say something like that. If gamma is very, very small, you would just get more or less the straight line and a little wiggle on top. Does that make sense? So this is large gamma. This is low gamma. I've now added up this straight line with that and drawn the whole thing. So this is really dq by dt. So if I had written dq by dt was some function of q, these curves are now this function of q. And we can do what we did when we were looking at a self-activating or self-inhibiting protein. We had some function of A and we were drawing those and we were seeing where do they cut. Here, what do I need to figure out the steady states? I need to figure out where is f of q 0. So where does it cut? The x-axis. And where does it cut? Well, here. These are the points it cuts. There's only one. And can you see that there can only be one? No matter what values are used for this and what values are used for that, as long as the switching rates were positive and gamma was some positive number, there's no way you can have more than one cut, right? So there is only one steady state possible. So we've already proved that if these parameters are all constant, you can't have bistability at this population level. What do we want for bistability? We want, for a given set of parameter values, we want there to be two solutions for the fraction of cells that are quiescent. We want the system to be able to stably sit in, say, a 90%, 95% quiescent state, where 95, 90% of the cells are quiescent. And we want it to be able to sit stably in another state where, say, 60% or 50% of the cells are quiescent. That's certainly not possible here. So now we need to add something. We need to clearly, if these are the only terms we're allowed to have, the only way to get multiple cuts is to make one of these parameters a function of q. Correct? I can't think of any other way. If we don't introduce any further elements, the simplest element we can introduce is to make them a function of q. Okay, so I'm going to continue with this by saying, and bear with me for a bit, I'm going to, for completely other reasons which I hope I will be able to make clear to you later, I'm going to assume gamma is very small. Okay, this has something to do, I'll just throw it out there right now. It's something to do with the fact that I want two different time scales. And because remember, we wanted to make an oscillator which has two time scales, and I want the growth to be the slow time scale. So I will come back to this point again, but if you accept this for now, I want to get rid of all of these possibilities and just concentrate, therefore, on very low gammas. So more or less the straight line itself. Gamma is not going to be very important. I want to create the bistability by messing with these switching rates. Okay? If I do not come back to this point later, please remind me. Please push me to explain this. But for now, bear with me. and let's examine VQG and VGQ and ask, can we change them? Can we make them some kind of functions of Q such that we can now make this line cut the x-axis in 3 places? Okay. So there are infinite ways to choose functions of Q, right? So I am going to narrow this down, and I'm going to say, let's assume these are switching rates. Let's assume, what does it mean for a switching rate to depend on Q? It means that somehow, this is the way I interpret it, somehow there has to be some communication between the cells in this population. The cells that are in the Q state and are switching somehow have some idea how many other Q cells there are. And they're changing their rate of switching depending on whether the cells are in lots of cells are Q or very few cells are Q. Okay? So I interpret this, there may be other interpretations of how a switching rate can depend on Q, but I interpret this as some kind of quorum sensing system. There is some quorum sensing system, maybe the cells are secreting something when they're in the quiescent state or in the growth state, and by looking at how much of this molecule is out there, maybe a cell can somehow adjust its switching rate. So how would all this happen? This would happen through some complex enough internal molecular machinery. We're not going to go into that. We're going to stay at a very abstract level and just say, let's assume the rate somehow depends on the quorum, on how many cells are there in the Q state. So I'm going to simplify this by saying, let's take a very simple version of it and see how far we can get. Let's assume that there are just 2 rates. There's a certain rate at which you can switch if there are very few Q cells, and a certain rate if there are a large number of Q cells. So let's assume a step function. Let's assume let's say QG versus Q is something like, I don't know whether I pick one way or the other way, but let's say it could be a function like this or a function like that. And we can pick where it switches and what level it switches to. All that's under our control, but let's stick to this and see how far we can get. Okay? So now, suppose I was to do something like this. I was to allow VQG to take 2 different values, and for the switching rate to step between these values. Could I get a line like this to cut the x-axis in 2 places? And the way I'll do this is the following. So let's get rid of this. This was the case where everything was constant. We said there were 2 values. We're going to allow some kind of VQG low and a VQG high. So let me draw out 2 values. So let me call this VQG high and some other value here, VQG low. So suppose it was only this or only this, these would be the straight lines. This is a constant. This can be this value or this value. But suppose it was only a flat value, these would be the straight lines that would describe these terms. By making it switch from one value to another, what I mean is that you are going along one line for a while, then at some Q value, let's say this value, some K, you are switching from that line to this line. Does that make sense? Can you see what I've drawn here? Is it big enough? Should I draw it bigger? Can everybody make out what I'm trying to say here? Let me draw this part in another color. So there is a notional straight line, 2 lines that I've drawn. This is the line that these 2 terms would follow if VGQ was whatever it was, this value, and VQG was the low value. Then it would follow this line. If it was the high value alone, it would follow that line. But if it's switching between those 2 values at some value of Q, then it has to start on one of those lines and switch and go on the other line. Now we want to make that cut in 3 places. So it's never going to work if you start here and switch down and then go like this. You will still cut in one place. But if you do it the other way, it can work. So suppose I went this way. Suppose I started like this, and I went down, and I switched up, and then I went down. Then do you see that you cut in 3 places? So I've ignored the gamma term, but if you want to include the gamma term and it's very small, just imagine the whole thing being slightly bent downwards. It would still cut in 3 places, as long as gamma is not too large. So let's assume gamma is not there. Let's assume gamma is close to 0. Does that make sense? So what does this mean? This means a particular choice here. So not all possible values of this will work, but at q equals 0, we are starting on the low branch, and then we're jumping to the high branch. So what this means is that you are starting low, and your switching rate is jumping up at some value k, which is exactly the value here. Q is equal to k. Yes? No, I'm working with QG. So if I got this right, then at Q equals 0, sorry, you're absolutely right. I have messed this up completely. So let's go back. Let's go back and do this carefully again. At Q equals 0, this term is 0, and it's GQ. Thank you, Vidya. So let's draw this properly. This is VGQ, some particular value, which I'll call low, and this is VGQ high. And this is VQG. And that makes sense because at Q equals 1, this term vanishes and this term remains. So what we are trying to change here, therefore I have to change all of this. What we are doing over here is having VGQ low, VGQ high, and VQG is some constant. We haven't really messed with that because that's always been the same. Thanks, Vidya. Does everybody follow this? So by starting off with a low G to Q switching rate, but then switching up to a higher rate at some point, we could potentially get bistability. Let's just quickly check that this satisfies our stability issues. This line, the yellow line I've drawn, assuming gamma is very small, is exactly dq by dt. It's the right-hand side. So when this is larger than 0, then you will be moving to the right. Let me use another color. So when it's larger than 0, you will be moving this way. When it is less than 0, your arrows will be pulling you that way. So arrows this way, arrows that way. And now you can see that this is a stable point, this is an unstable point, and this is a stable point. It's a familiar pattern that we've seen. There are 3 cuts, there are 2 stable points, and 1 unstable in between. So in principle, if you had all of these processes, and you made VGQ a step function of the kind that I have drawn over there, then you could get bistability. Then it means keeping all these constants the same. We're not changing this value, this value, this value. So how many constants are there? How many parameters? There's 1, 2, K, 3, 4, and gamma. 5 parameters. For keeping all those 5 values fixed, depending on whether you start with a small number of cells in the quiescent state or a large number of cells, in the long run, you will end up in 2 different steady states at the population level. Yes? We will come back to that. Right now, so this is exactly the next step we will talk about. At some point, I will also point you to Sunil to talk about that. But we will talk about whether we can keep gamma constant, whether we can keep these other values constant or not. Because precisely the next step will be, once we've got the bistability, we will want to add some change in one of the parameters in order to get the oscillations, if you remember the frustrated bistability. Now we'll come back to ask which parameter is the relevant one. So hold on to that thought and we'll talk about this. Okay. So far so good. You can get bistability. So now let me get to the frustration part of the bistability. Is that okay? Any other questions about bistability? All right. So now let me tell you how to get oscillations. The idea is the same. You saw it before. All I'm going to do is exactly the following. I need to now have some parameter that allows me, if you remember the frustrated bistability case, we would now have something like, well, Q on the y-axis. We want some parameter on this axis, and we want some bistable branch, and then we want to make the parameter depend on Q so that you start going around. So I need to find some parameter that varies this curve in such a way that I can have monostability on one side, bistability, and then monostability on the other side. Okay, so I'm going to make one choice, and I'm going to still stay abstract, and I'm going to say, imagine there's some parameter that, let's say, changes only these values. Okay? So let's imagine, where's the yellow one? The yellow. So imagine there is some parameter I change, and I decrease this parameter. So the reason I'm picking this direction will become clear in a second. But let's imagine I change this parameter, and I can make this whole curve, this part of the curve, sort of go up. So I'm changing these values. What would a curve like that look like? You could imagine that it starts going up and here and comes down like this, but it does not cut. So if this parameter pushes these values high enough up, then you would get a situation where the only place you would be cutting is at the high state. Similarly, if you made it low enough, maybe, if we've set up the things correctly, you could have it still non-zero, but low enough that you come here. Well, I'm not drawing this well, but let's say you come here, you jump up, and you still do not cut, and you go down here. In which case, the only cut would be there. And in between, you could have a region where you cut in 3 places. It's exactly analogous to what I was doing earlier in the previous thing. So there is some parameter, and you will have a region where you'll have bistability, and 2 regions where you'll have monostability. Let me just say, and we will come back to why I'm going to do this, because I'm going to make a particular choice at some point to go back to the biology of what this parameter could be. But let's imagine that the increase of parameter pushes you that way. So this is going to look a little flipped over from what I showed you last time. But essentially, when the parameter is small, you only have the high state. And when the parameter is large, you only have the low state. So your steady states are going to look something like this. That's your bistable system. And now, if I made this parameter dependent on Q, I'm still keeping it abstract. And I put in these 2 different time scales. What would the oscillation look like? The oscillation would start here, would creep this way, would fall off the edge, creep this way, and fall off. So the oscillation would go like this. Okay? Let me draw out what it would look like as a function of time. That's Q. Let's say you started high. You start at some high level. It sort of drifts down a little bit, but slowly. Then it has a very large drop and a very fast drop in a very short amount of time. And then it's here, and then it goes up. And then it goes up like that, drifts down, goes down, just like we drew in the previous class. So something like this could work. So now, all we've done so far is go from what we did last time, which was more in a molecular context and thinking about gene molecular networks. And we've just said you could do exactly the same thing. The whole idea was so abstract that even at a population level, if you picked and chose correctly, then with just switching between states and growth in one of the states, but no growth in the other state, you could set up a population model that has all the properties. And if you pick this parameter correctly and you put in all the things, you could get this oscillation. Right. So that's as far, to some extent, as one can go abstractly. At this point, it starts to become important to get back to the biology, because now we need to ask, is this a sensible model? Does this say anything about the biological system at all? What can you do with this model that would be useful for Sunil? Can I make some predictions? Can I tell him, here's an experiment you can do and go back and look at, and maybe this is what you should see? How do we build confidence in this model, as opposed to the 100 other models, or the infinite other models that Arno rightly said could equally easily give oscillations like this? So we have to start now connecting back. And this is the point at which I also started sort of talking back again with Sunil and saying, hey, I've got all these shapes and I've got all the things. And I must admit, at one point, I spent a long time worrying about the shape of it. You see, this shape goes down and then goes slowly up and then up. But if you look at some of the things he's drawn, it's the mirror image. And now we know, of course, that that depends a little bit on the strain. There are strains which do exactly that. But at that time, I hadn't appreciated the point that there were strains that do this and that and show all sorts of waveforms. And I was very worried that we were getting the wrong shape. And that was nevertheless useful, because one of the things you realize, what would you need to get the other shape? So suppose I wanted it to slowly go down and drop, but then do a shape like that. Maybe I should draw it in another color. Suppose I want the mirror image shape, which drops fast, but then keeps dropping and then goes up. Notice that in this framework, all you need to get that kind of thing is to mess with the type of bistable branches you have. If my bistable branch was like that, that would be enough. So it gives me a way of thinking about how do I get different shapes. That's one of the uses of a mathematical model and a mathematical framework. Even though it's abstract, it constrains you to try and figure out how to get different kinds of shapes, different kinds of things, and it points you in certain directions. So I had to start, and I spent a long time thinking about how you would get that shape. I won't go into the details because it turned out to be not as easy as I thought, but in the end, not as difficult. And I simply needed to make something that was where not just the two values that it was stepping between, but it was a slightly more complicated shape. So we won't go into that because, in a sense, that's not, now I realize that's not so important, because there are strains that actually show this also. And if we want to get into the details of why one strain shows this shape and another strain shows that shape, then this question becomes important. But for now, let me leave you with the thought that actually multiple shapes are possible. And the only thing, when you separate timescales, that determines the waveform and the shape of the oscillations you see is really these original bistable branches. Are they going up with the parameter? Are they going down? And so on. Okay? And one can then start to think about it. But what we need to really connect to the biology now is to ask what this parameter could be. What is a parameter that could make, so what have we done? As the parameter increases, the rate of switching from G to Q decreases. So now we need to think about real biology and ask, what parameter could make the switching rate from G to Q go down? Any ideas? I think Sunil has already given it away slightly in one of his slides. So if you were looking, you might have seen it. You want to make a wild guess of what could be happening? What is the thing in this chemostat that could make the switching rate from growing to quiescence reduce? Nutrients. Right? That's perfectly reasonable possibility. I knew that he was introducing glucose into this chemostat. So in my mind, before even talking to him, I thought it's glucose. So for the rest of it, in my mind, I was thinking of this parameter as glucose. Later, when Sunil takes over very shortly, he will maybe talk a little about this. And he'll tell you that it's not glucose. He has other ideas of what it could be. Could be oxygen, perhaps. I don't even know. So I never thought about oxygen, but I don't know what the biology of yeast is. Would it switch? But exactly. I mean, this is exactly correct. this is the next step. Thinking of all the different kinds of things that could be happening and what would they be doing to all these switching rates. Let me go with what I thought, which was glucose. So I was thinking of nutrients, and I was thinking of nutrients, presumably for the same reason you mentioned it. The more the nutrients, maybe you only switch when you have low nutrients. Maybe this push to go quiescence and a transition to quiescence happens when somehow food or some metabolite goes below a certain threshold. So we could pick that and we could try and figure out precisely how this nutrient would change in the system. That would mean exactly the following. So what we have done is we have already made this into a step function. So we made this into, the other one. This a function of Q, we added that. And now I will also make this a function of another variable A, and that will be the nutrient. And I will have to write down an equation for this. And I will put in processes like supply of the nutrient. So let me call this the nutrient per cell. So this is nutrient per cell. And there will be things like supply. This nutrient, you could imagine this both as an internal cell, inside a cell, a nutrient, or even something that's accumulating outside. It turns out it doesn't matter much. But either way, there would be dilution. So you would have terms like this, where you would have a dilution rate, which we know is something like that, multiplied by the amount of nutrient. You would also have consumption, presumably. Maybe if you are a growing cell, you're not only, the nutrient is not only just diluting and going away when a cell that contains it is removed, or if it's externally when it's diluted out, but it could also be consumed. So you could put in something like another consumption term, which would also look remarkably like this, because it depends on the number of growing cells. This would be something like a parameter that tells you what's the ratio between dilution due to outflux from the chemostat and what's the consumption rate. So you could put in these kind of parameters, and we could do all the things that we did, which was to take a system like this and now call this glucose or A, and then ask what is dA by dt, and does it go through this? This would be dA by dt equals 0. All the kinds of things that we've been doing. Does it fall through? Will the oscillation happen? And so on and so forth. Why is this useful? Because it starts to now get towards certain kinds of experiments that I could perhaps go back and tell Sunil to try. For instance, the parameters that show up in an equation like this, once you're really starting to think about a real nutrient or a real molecule, these are parameters that he can control in his chemostat. So I could now investigate in this model what happens if I vary this supply rate. Okay? If I actually draw this out, so let me go back to this. This was the bistable thing, and if you remember, we had to draw another line which was dA by dt equals 0. It turns out this line will actually look something like that. It actually looks a little steeper than this. It goes like that, it goes off to infinity. And if this crosses in between the two branches, you can get an oscillation. But what happens if I increase sigma? Well, if I increase sigma, the line gets like that. It gets steeper. And at some point, it will hit this. And if you remember, I told you that when it hits that branch, you stop getting oscillations. So I can now predict for Sunil that if you start tuning up the amount of glucose you give, you will stop oscillations. You will not get oscillations beyond the critical value. And I could try and predict the critical value. And if I predict the critical value, I can do a little bit better than that also. I can also predict how you would break down the oscillations. What would the oscillations look like if you were oscillating and then you just slightly increased the supply rate and ended up with a stable thing. So imagine, it depends where you are in the oscillation. Imagine you're somewhere here. Then you would still creep down, you would come down here, creep up, and only when you hit this would you stop. But suppose you were somewhere already here, you would just creep up and stop. So depending on where you are in the oscillation, that's a prediction I can go back and tell Sunil. Why don't you do this? and exactly at different points in the oscillation, increase the supply rate and see what happens. And I predict that in one case, you will actually go through a whole cycle before you stop. But if you start too early or too late, then you will just stop very quickly. So I can start making predictions of these sorts. You can also predict the same thing when you reduce the supply rate. You will actually go and hit over here, and you will find a steady state at a different level. So these are predictions you can go back and make. And this is the level I was thinking at. I'm going to stop here because when I went back, there were two things that I think I was excited about in all this. One was, of course, to make predictions like this in order to get more confidence about the model, to understand whether this model is capturing enough features of the real system. But this was immediately a way I could think of where Sunil could go back and test the model in some sense. So it's important to predict something that can be tested. But the other thing that was exciting me was something a little more abstract, was right from the beginning the question that in order to get the bistability in the first place, if all this story happens to check out and is true, it was important to make this a nonlinear function. We could not work with a linear function. There were other things I had tested. You needed some nonlinearity, and you needed some dependence on Q, which I interpreted as a quorum sensing system. So I was very excited that maybe he could go back and he could check whether these systems are secreting something, are they quorum sensing, are they doing something else? That for me was very exciting. When all this conversation happened with Sunil, he noticed an entirely different thing, which I probably would not have noticed, which is a very obvious thing, which is what happens to this resource along this oscillation. So I had told you that I can get various different kinds of shapes now. I had ways to get this one, the fast and the slow rise and the fast rise, or the fast drop and the slow drop. I could get all sorts of shapes. But one thing that was common to all of these shapes, if you draw out the resource as a function of time, the resource always builds up just around about the time where the large transition happens, or just before it perhaps in practice. It starts going down, and then it goes up, and then it goes down, and so on. So the resource also shows oscillations, but it has a very particular relationship in time with the oscillations of the number of cells in the quiescent phase. And that seemed to be independent of all the waveforms. No matter what the waveforms, it didn't really matter whether this was like this or the mirror image or flat, all sorts of things. This was always showing a peak around about the time of the first drop. And this is something that Sunil noticed. And he's interested in metabolism, and he was interested in not those abstract questions I was thinking of or the more specific predictions I was thinking of, but since he's interested in metabolism, he was wondering, what is this resource? And he didn't think glucose was the resource, or whatever intuition he had. He wanted to find out what is this metabolite that's driving this, and thereby open up a whole set of questions about which metabolites really affect this growth, proliferation, transition. So I will now switch over to Sunil, and he's going to tell you how basically he took this one observation that the resource was peaking right before to try and investigate which resource. So I'll just leave you with the thought that, in many ways, the model does various things, and you can, of course, you should make predictions, but sometimes you raise questions in completely other directions. And questions that occurred to me from the model and from seeing the model were different from questions that Sunil thought of. And that's why every once in a while you have to start communicating with each other and then decide which question is the most interesting. So I'm still hoping that he will do experiments to check the model in more detail. So eventually at some point when I give a talk about this and somebody asks me, what about another kind of oscillator? What about this? What about this other model? I'll be able to say with more confidence, no, we've checked this, we've checked that, we've checked this. This is the model. But at some level, also, I don't care. If it's raised a nice and interesting question for Sunil to look at, maybe that's the purpose of the model. So on that note, if you have any more questions, please tell me while he's getting set up. But otherwise, I'll leave you with that thought. Yeah.
Was there a, hello? Yeah, so was there a reason to take VQG constant and not the other way around?
You could do the same thing. So you could do the other way around, you could make this 2 different values and this change, and that would also work. You could make both of them step functions, you can make that work.
So
you could also make them not so steep, but more like hill functions, that works. Essentially, there's a lot of things.
my question is, why did you choose this? Like,
Not any particular. You can get pretty much the same sets of things I'm showing by the other way around. So we don't know. We don't know. The model at this level is not going to be able to answer the question whether this resource, whatever it turns out to be, is only changing the G to Q or the Q to G or both or neither and something completely different. Maybe there's something else going on. Okay? All right.
So this was really nice in that what Sandeep could do without knowing anything about the system is pretty much get exactly this pattern with the right proportion of quiescent cells, growing cells, quiescent cells, growing cells in the oscillation. And what it pointed out, really, however you slice it, is that there has to be this oscillating resource in the system in order to maintain this exact kind of phenomenon. Okay, so now this is where it becomes very, very interesting to me and also fairly general, because this might tell us how cells can, kind of a sufficiency model for how cells can switch between quiescence and growth, which is a very general idea for cell biologists, right? So we talked about parameters. This is a chemostat, okay? So this is the system. It's very well mixed, okay?
Does the chemostat have a tap?
So I'm drawing that out. I'm going to draw out what it looks like. So here's your liquid, let's say 1 liter of cells or something like that. What you have is a little pipe feeding in media, and this is constant, okay? So we set this at a constant rate, which means it's feeding in continuously all the time. And to maintain volume, you'll be taking out the same amount of liquid at a steady state. And this is steady state. So what we're not doing here is adding a bolus of glucose every half an hour or something like that. This is steadily flowing in. So it's a really steady state system. So inside this, if you just look at media glucose, it's pretty much going to be like this. It's flat line. In fact, it's very low because as soon as we add glucose, it's taken up to the cells. So it's almost non-measurable somewhere here. Okay? Oxygen now, this is a well-aerated system. So there's a pipe constantly being fed in. But what we're reading out there are these oxygen, amount of dissolved oxygen in the media. So despite steady state oxygen available constantly, oxygen is oscillating because that is the pattern we're looking at, this one, right? But oxygen itself then can't be the resource that's controlling it because what you're feeding in is constant oxygen at whatever, 1.5 psi or whatever. Can't be oxygen. So it has to be something, presumably, that's coming from here, that the cells are building up. At some point, as it builds up enough of this metabolite should be able to control all biochemical and genetic events that the cell needs in order to switch to a growth state. But in the process, this metabolite has to be consumed because that's what's happening to the resource of the metabolite. It's going back down this way. And so that then will send it back up there to the Q state, because the moment it goes below this threshold now, as it's committed to growth, it has to go back there. So it has to be some kind of an oscillating consumable resource. Now the good thing is we had a lot of metabolite data. We have data for over 100 metabolites taken across this cycle, right, at every point of time. So we have sampling across like this every 20 minutes or so. So we know what metabolites oscillate in each phase of the cycle for about 100 metabolites. So now, so we'll see what the model predicted. In this case, I did a slightly kind of a data, let's see if data fits the model kind of an experiment. So this is fraction Q, right? This is some time. And Sandeep's model is something like this, okay? And then his model predicts that the resource usually goes up change the color here. The resource goes up like this, comes down like this. Okay, so here you have resource. Okay, that's the resource, and this is the fraction coefficient, and this is what his model predicted. We have data for over 100 oscillating metabolites. So we could look and see what could fit or be close to what happened here. What we saw was actually rather really nice. Despite having all this metabolic data, so now we'll superimpose, these are our actual oscillations. In this case now it's dissolved oxygen, which is representing Q and G state. And in all of this, only 2 metabolites actually had almost exactly the same pattern. One was actually perfect, the other one was close. That's it. This is all we got out of 100. Now this metabolite, perfectly oscillating metabolite, turns out to be acetyl-CoA. And this metabolite, the smaller one, was NADPH. Okay? Might mean nothing to some of you, might mean everything to some of you other guys. Yeah? Those of you who work with biology. I'll tell you why now this makes perfect sense to be an oscillator. But we did one more thing. We drew a kind of a phase diagram. Maybe I can draw it here. Okay. So we also just represented this slightly differently as a phase diagram. So instead of plotting it this way with time, we did fraction Q with resource cell. This is what Sandeep's model predicted. And in this we got this kind of a phase diagram for what the resource should look like. I now replotted acetyl-CoA, which is what this plot looked like, to see what it looks like over here. Now we looked at the change in, so this is still dissolved oxygen, yeah, and this is change in resource acetyl-CoA. As we looked around, and we pretty much got exactly the same phase diagram. So it's really saying to us that, making a parsimonious argument, we have all these metabolites, only 2 more or less meet these criteria, and 1 almost perfectly meets this criteria, it's acetyl-CoA. So now, can we understand from all of this though, how or why this metabolite might be the resource that is controlling this oscillation? So if you want to have a resource that controls oscillations, it needs to have, biologically, the following kind of properties. A, it has to come from something that's being fed into this system. It can't come from thin air, right? Now, we know experimentally, this is what we had done over many, many years and hadn't made enough sense out of it. But the way you could control the oscillations is by how much glucose you had. You had a few other things that could change the oscillation. So in our case, we could add acetate, we could add ethanol, acetaldehyde, in a few cases, we could add amino acids like glutamine. Okay, so all of these could reset the system. Now the beauty of all of this is, regardless of what these are, in order for them to be biochemically useful, they all break down to acetyl-CoA. So glucose goes to acetyl-CoA. Ethanol goes to acetyl-CoA through a slightly different route. Acetate, acetaldehyde goes to acetyl-CoA. Glutamine comes this way, goes to acetyl-CoA. All of them go to acetyl-CoA. So regardless of where you are in the spectrum of things we know can control the oscillation, you go to acetyl-CoA. What does acetyl-CoA do in order to be able to control the system? This is an unusually special molecule because it can do multiple different things at the same time. If you want to commit to this growth phenomenon, you need to make biomolecules for growth, primarily things like lipids. you use acetyl-CoA to make fatty acids. You can't get fatty acids without acetyl-CoA. We also use acetyl-CoA as a source of energy, which is something you need over here. Acetyl-CoA enters what's called the TCA cycle to make ATP. We already know from our data that this is the maximum growth phase. The maximum amount of respiration to make ATP happens here, and the maximum amount of energy consumed is also over here. This satisfies the criteria to make energy. It also has to have all those gene expression things that we see in the noisy gene expression profiles. And acetyl-CoA does something amazing there as well, because the main way cells use to regulate gene expression is by acetylation of histones. Okay? And people study acetylation of histones as a post-translational modification to activate gene expression, forgetting the fact that the only way you can get acetylation is by using acetyl-CoA as your acetyl donor. You don't get acetylation without acetyl-CoA. And so if you build up enough acetyl-CoA now in the cell, it can go into the nucleus, increase acetyl-CoA amounts in the nucleus, acetylate histones, and then there's beautiful data experimentally that we, Bentu's lab had, which showed that as acetyl-CoA amounts increased, you had specific histone acetylation in regions where there were the promoters of genes correlated with growth, like ribosomal genes and so on and so forth. You could observe experimentally this pattern of histone acetylation, activation of gene expression for growth, and then see all the genes that we know from this huge gene expression data profile that we had that come up and go around in this same fashion. So we can now make a fairly strong parsimonious argument for why acetyl-CoA could be a regulating resource. And the idea you have now is that you have cells. Where is the oscillation? Here. You have cells. Now what we know over here in this Q kind of phase is that they're building up storage carbohydrates from all the data that we have. They build up to a certain point over here, which means now you have enough to liquidate. When you liquidate, you start making acetyl-CoA. So acetyl-CoA is built up as you're steadily making and liquidating it. It's reached a certain threshold. This threshold amount in that cell is enough to flip it over to a growth state, because there's enough now to carry out both fatty acid synthesis, ATP generation, and gene expression activation. But as you do that, you have to use up this resource for all those processes biochemically. So you start going down again, which then means you go below this level and you'll have to flip back to your original quiescent state. The other metabolite, which was close but almost there, is NADPH. This is again a perfect thing you need coupled to acetyl-CoA, because you can't complete your growth process without this molecule. What this does is provide all your reducing equivalents that you need in a cell for all the reductive reactions. To make the biomolecules, you need to complete growth. So many of the, along with lipids and all of that, many of the amino acids that you need to make here require ATP. You need a whole range of reductive sulfur metabolism to happen later, which requires NADPH to complete its process, and so on and so forth. So you can actually now make a very nice biochemical parsimonious argument on how just the oscillation of this resource within a cell in a population can result in this oscillation. And so many different things in the model suggest that this is quite possible. Okay, so there's a small limitation here as experimentalists. So this is still coming from the bulk data that we have. What would be ideal is if in real time we can see this happening in this population of cells, to see acetyl-CoA build up in one cell above the other cell and so on and so forth. There's no way to experimentally do this right now because we don't have any in vivo visualizable sensors for acetyl-CoA. What we can only do is collect populations of cells, measure it. We can't sort out the cells and measure because metabolites change very quickly. So we can do that with gene expression reporters or something. We can't do it with metabolites. But it's at least telling us this is what we need to look for, and given us a very nice way to explain what is happening biochemically using a very simple single kind of a molecule that can set up all the other noisy data that we have from years of study that's present in the system. Yeah? So that's really what we have right now. So we can now go back to this question. The number of, it's only some cells dividing in each cycle. What we can propose from this system is that in all cells, at any given point of time, acetyl-CoA is building up. From this model and a few little things that we can tweak in the model, we'll basically come to this point where if acetyl-CoA reaches this threshold, you'll commit to the growth profile. So those cells are now crossed the threshold, they're committed, they'll complete growth. But now in that one cycle, they'll use up so much acetyl-CoA that in the next cycle, by the time they're ready to grow again, these are probably at the bottom of this pool of cells, while the other cells, which had not used it up in that one cycle, should be in a position to enter the next round of division and growth and so on. So you could have this system of constant oscillation between growth, quiescence, growth, quiescence, growth, quiescence. So that's the argument we're trying to make over here. So what we have is really a fairly simple, this is really a simple kind of a very large, from looking from far up kind of a model, coarse-grained. It is very coarse-grained to say what might be possible. What we've been able to do now, because of this very simple model, is constrain possibilities to this space and explain or start thinking about how this metabolite mechanistically might be able to control these kind of events. And there's very interesting, nice, separate biochemical experiments showing how acetyl-CoA is the central molecule for growth. It's essential in cancer cells and this and that and all that. So it all starts to make sense. So I guess the point of this whole exercise was to see how we could reduce these very complex, noisy biological data systems to something that is now very clean, which we can measure quite nicely and actually say that this might be causal in carrying out a switch or a transformation from one cell state to another cell. Yeah? So let's stop with this part over here. Yeah.
So there is this difference in gene expression in a colony itself. There is heterogeneity in a colony itself, right? So is this something has to do with that?
So we have some time. We can talk about a colony if we want after this. We'll take questions on this though. Important thing over here is this is a very, very well mixed system. Okay. So in this case, there is no spatial separation. What you have in a colony is a static scenario, very slow diffusion of nutrients and so on and so forth, which will change what is going on over here. Here now, in a chemostat type of system, whatever is there internally is equally accessible to every cell at any given point of time. There is literally no gradient that can happen. Yeah. The only way it can happen is internally in a cell, the rates of utilization of a molecule or buildup of a molecule at any given point of time are different. Which goes back to yesterday's talk of how you can get internal differences in biochemistry and set up a stable oscillating system. Yeah?
What makes you...
No, you can go ahead. Pass the mic there.
I want to ask, so what makes you think that it is not density-dependent effect?
There is a density-dependent effect. This is a great question. So the question is there a density-dependent effect over here? What we know experimentally is that we can see stable oscillations only at high cell density. We know that. If we have very low cell density in the same system, we don't obviously see oscillations. What we still don't know is whether that's a limitation of our experimental approach, because what we're measuring is some bulk readout, dissolved oxygen, and not something else. But there seems to be some clear experimental dependence on the high cell density, because when we now put them as single cells that are separated without the ability to kind of interact or communicate, we don't see steady oscillations when you look at least something like a gene expression reporter or something like that. So it breaks down. We haven't quite addressed density dependence, right?
I mean, in one sense...
Except we kind of do, yeah.
In one sense, the fact that the switching rate was dependent on Q is also a sort of density dependence.
Yeah.
So there are certain density dependencies that one necessarily needs, but whether there's some other processes that are also density dependent, maybe we don't know.
Yeah. So what we've got is an implicit assumption, since it depends on the number of cells in Q, that there is a density dependence, but we haven't fine-tuned what this density dependence is, right?
No? Yes? But it's only about density, or if it's about like confinement?
Confinement. So in this scenario, there's no confinement because it's perfectly well mixed and everybody is moving around at a very high rate at all times. I don't know how much time we have. Another 20 minutes, half an hour, 15 minutes. I could talk about a spatially separated system if we have time, but we can see if we have a lot of questions, we can stop here. Or we could breeze through some spatial organization where some entirely different rules come into play.
Yeah. So, is there any parameter in the model that if we tweak with that, that changes the percentage of cells? Actually.
Well, the percentage of cells depends on the resource, right? Which we can easily tweak.
So in experiments
the model predicts
also,
as you increase the resource, you'll break it down into, and if you decrease the resource, also you'll break it down. This we kind of know experimentally. If we increase the amount of glucose, the oscillations become shorter and shorter, and then break down. But also if we decrease the amount of glucose, which will lead to this how much acetyl-CoA is there, again it starts becoming shorter for different reasons because of the breakdown of the bistability, and then you don't get oscillation. That is implicitly, I might have erased that part, but it's implicitly there in the.
So you mean if you increase the glucose or the nutrients, the percentage of cells that goes to growth phase
what will functionally happen is now all cells will have a steady supply of high acetyl-CoA. So what will happen is the Q parameter. Where is that? Here. The number of cells in Q. This one will start decreasing very dramatically. So it drops very quickly here. So this starts getting smaller and smaller, and then it's gone.
Maybe I can just add one thing to that, is that clearly from the model, if you just look at the model, forget about the real system, in the model, it's that original bistability that really tells you what's the upper level of the oscillation and the lower level.
Right.
So in the model, if you need in the model to make that change, and you want the 35% to become 50% or something, you have to go back to all the parameters that were affecting the original bistability. They must be changing. Now, is that happening in the real system or not, and in which way they're changing is partly an experimental question, one of the things one can test. But within the framework of this model, the shape of those bistable branches is what sets those two levels. So any parameter that can change that will change the numbers. And any parameter that leaves those unchanged will leave those numbers unchanged in the oscillation.
There's a question back.
Hi, can you hear me? Oh, okay. Couple of questions actually. One was, I'm forgetting the first one. Let me go to the second one. Since, oh yeah, I remember the first one. So this is with regard to...
It's in a bistable state.
It's a bistable state, no?
Yeah, acetyl coenzyme A. So, since, without even getting into the fact that you're seeing changes in quiescence or oscillations between these two cellular states, you are seeing an oscillation in the concentrations of, say, acetyl-CoA
bulk.
over time, bulk. What do you think might be entraining the acetyl-CoA concentrations across cells over time?
It's internal, right? So
it's internal. So how are these internal oscillators being synced?
Yeah, so there's a little thing, I don't know, but I'll speculate. So to start these oscillations, essentially what you do is you initially have a batch culture. You use up all the glucose in the system, you starve the cells, and then you feed glucose. This is how you get the oscillation in the first place. If you're starting off with feeding glucose right off from the start, you never get oscillation. That's because all cells have unlimited supply of acetyl-CoA. They don't really go, a large enough fraction don't go to Q. Here we are starting with exactly 100% starved, proper full Q state. In this now, if you just have, so now you're feeding glucose, they're all now starting to take up glucose and start their biochemical processes. We still can't exactly address how you start the oscillation, but what you know from biochemistry is that different cells will start accumulating acetyl-CoA at different rates. And if threshold is not too tight, if you put a very tight all or nothing threshold, then it becomes a problem. But if it's a, anything above X versus anything below X kind of a loser threshold, a population of cells will start going above and start coming down. And with a couple of rounds of cyclings of this, you might start getting stable oscillations. That's the loose idea. But we haven't really addressed how we've started this whole biochemical network cycle thing going on. It's a little tricky.
It's to, again, acetyl acetyl-CoA. And the idea being that since you have a limit cycle in terms of oscillation of acetyl-CoA, have you sort of spiked the chemostat with, say, glucose while the oscillation is on and seen phase shifts?
So that's exactly what happened when I said these are the things that change oscillations of phase. Now let us say you're here in this Q state. Over here, you're a long way from here. As soon as you add either acetate or glucose here, you'll immediately, within a very short time, jump right into this state. So that's how we know that these are the molecules that can phase shift, right? So glucose, acetate, acetaldehyde, glutamine, all of them can do it. Pretty much everything we put in that can do it, if you work back to see where it's going, all of them can work back to acetyl-CoA. So there's a 100% correlation. There are about a dozen molecules that can do it. Interestingly, all 12 of them at some point will go to acetyl-CoA, which is pretty neat. So just by, you're making this very strong argument just by saying, 100% of the time all these molecules go to acetyl-CoA. Reasonable chance that this is your molecule. Yeah, it's a parsimonious argument, but reasonably strong. Yeah.
Yeah. So fine. My question is to Sandeep. Basically, he was talking about small gamma is related to 2 time scales.
Thank you. I didn't actually come back to that. I said I would come back, and I never came back. So you want to ask about that?
Yeah.
Okay. No. So if you look here, we needed 2 time scales. I needed, if you go back to the frustrated bistability idea, you needed things happening at the Q level which cause the bistability to happen quickly. So process, generally the dynamics of Q to be fast and the dynamics of A to be slow. Within A, the kind of processes that come in are basically supply and dilution and growth. So I could not, in order to make this slow, I would have to make gamma and sigma slow in order to get the 2 time scale separation. If I made, therefore, I had to keep this slow over here. And therefore, this term never really had much of an effect on this dynamics. If I had made it such, I could have gotten, if you forget about the A equation, I could have gotten bistability by making gamma large and messing with gamma and making gamma dependent on Q or something like that. I could do that. You could certainly do that. But if you do that, this would have to be overall on the same time scale as this. And if that comes down here, then this would also be on the same time scale. And you can show you can't get oscillations. So there was no way of getting the time scale separation if I made gamma important enough in this equation. The only way to get time scale separation was to have all of these processes happening slow, and therefore gamma to be slow, and therefore it never really mattered here. So that was sort of the logic. Yeah?
It's okay? Okay, right. So I'm trying to link the 2 scales, the single scale and the population, because all the metabolism of acetyl-CoA, etc., is internal to the cell, and you said that to have oscillation, you need a high density. And so something must be released by the cell in the medium. So did you try to analyze the medium and not the cell?
So here's the experimentally difficult part, right? Especially the metabolite type of approaches. So what is relatively easy to do, what we do in the lab by mass spec and so on, is if we have a library of 100 or 1,000 molecules, it doesn't matter. If you know what their masses are, you can put it, train the instrument to detect them, and then in an unknown sample, see if those are there and how much of that is there. If we don't know what we're looking for, unbiased metabolomics, so to speak, that's really much harder, because what you're looking at are thousands of really complicated peaks, and then we'll have to look at the appearance of some molecule in these thousands of peaks over time, which is very hard to do, and then try to identify what that is chemically. That is one of the experimental challenges that we have to deal with as experimentalists. What we can do very well is look for known metabolites. So that's how we have this 100 metabolite data set. These are all the metabolites in central metabolism that, based on gene expression, we know should change. But unlike the protein space, which from genome you can go to genes to protein, We can't do that with metabolites. So in reality, what we know of are 1,000 metabolites, but almost everyone predicts somewhere between 20,000 and 100,000 metabolites in a cell. So we actually know, we're looking under a light bulb because the light's falling over there. The unknown space is very, very large, and it's not easily predictable, even by chemistry or so on and so forth. So that's a challenge. Separately, that said, though, we have looked for what you might think of as quorum sensing molecules in yeast and so on and so forth, and nothing obvious has come out. So you have a few Uranus type of molecules that are known to maybe control quorum behavior in yeast. We didn't find those, but that doesn't mean something else is not there. We didn't know what to do. But this is, you're right, there has to be something that entrains or sets up the rhythm, presumably a molecule that's put out. Alternately, it could just be the number of contacts, or by density, you can have things that come from physical interactions. The number of interactions you have per unit time in a high density culture is going to be much more than a low density culture. And those kind of mechanical, physical properties might also be entraining a system, and that we don't know. So we're actually trying to think of how we can sort out or separate the two. I think it's one of those going to be those long work in progress things where we go back and forth and try to figure it out.
And using mutants, no, there are not different strains of mutant. And
the nice thing is, experimentally, separately, people have shown that like the key acetyltransferases in the SAGA complex for histone acetylation, those would completely kill the oscillation. There are other things that kill the oscillation for obvious reasons. You can't enter growth, you can't enter the cell cycle. So you can really mess this up very quickly and make very wrong interpretations, which is what we tend to do as biologists, make a grand interpretation that's wrong. This helps to constrain. But in this case, precisely though, we could show, people have shown that acetyltransferase mutants, specific ones that should work at this concentration threshold, can result in either lengthening or breaking the oscillation, which then becomes consistent now with what we're seeing from a model.
I presume that the oscillations that you see here are also linked with other pathways in the cell, for example, glycolysis. So the glycolytic flux must be oscillating?
Yeah. So what we can see...
Can I just finish that? And perhaps there are also respiratory oscillations?
There are.
So which means that there are other known feedback loops which are interacting with this, and they are intrinsically capable of setting up oscillations on their own.
Correct.
So I'm a bit uncertain about how you factor them out.
So we don't factor them out, except that we've still said we've not figured out how you start the oscillation. We can see, this will kind of tell us how we can maintain this oscillation. But what we see from this, both gene expression and metabolite profile, is that you have to build up internally a carbohydrate reserve somewhere here. Glycolysis is highest over here, just as they start committing to this switch. This is the respiratory phase. So you're following glycolysis by respiration, and that happens only when you have acetyl-CoA. You can't enter respiration without the acetyl-CoA buildup. So this is a prerequisite to enter the TCA cycle. And then here you switch off respiration as you enter the cell cycle. One argument that's been made is that during the time of DNA replication, you don't want too much replication because you want to prevent DNA damage. So that's the argument or logic that's been put out. So this is this very clean cycle, glycolysis, respiration, very low respiration or glycolysis, but you've built up enough, and you start up building glycolysis slowly. So you have these two oscillations that are going on: glycolytic oscillation, a respiratory oscillation, and a shutdown of both.
So, but the point I was trying to make was somewhat different, just to get your comment, which is, what if you take the point of view that you have primarily a glycolytic oscillator here, let's say driven by positive feedback through phosphofructokinase, and then the phenomenon that you are observing here is a secondary consequence?
That's possible. And this is something I've thought of, and it's going into this, I can't separate chicken and egg over here. This is entirely possible, and that can happen.
So maybe I can make some comment there. One way perhaps to distinguish the two cases would be to ask what sets the time period of the oscillation.
Yeah.
In the case of the glycolytic oscillations, then it would be set by some internal parameters of the glycolysis, and mutants there would change the time period and so on, and one could test that. In the kind of more population model we have, the time scale is really set by the time of build-up of the resource. The slow time scale is what sets the
fast time.
The thing. So the build-up and the consumption, the slow processes are what set the time scale, and therefore that may be a handle on trying to figure out which is going on.
Right, and there's something else interesting, which is that there have been detailed measurements on the glycolytic oscillator involving injection of glucose, involving measurements of phase shifts, phase response curves. So there's a body of literature available which could be used to sort of test against the predictions of this model.
That's right. This is actually true. With the variation, and this is actually a very large body of work, with two kind of variations though. The classically studied glycolytic and respiratory oscillators are not in this time scale. This is like a many hour time scale. Those oscillations are much shorter in the minute time scale. And B, in this case, unusually in this system, we have a steady state constant supply of glucose. So it is not a kind of batch system where internally you're building up and using only. Here we have a two-layered thing where outside we're providing constant, steady, low glucose at any given point of time. So at time X, cell has glucose as a resource constantly available, but the variation in glycolysis comes from the buildup of other carbohydrate reserves which come from gluconeogenic pathways. And that is a confounding, not a confounding, but that adds a level of complexity so that we can't we can't really separate out how we're, but what we do see is this burst or liquidation of carbohydrates here. So there is this increase in glycolysis. There is this is happening both at presumably biochemical levels and at gene expression level. There is this higher order, much deeper problem that we do have to address. We haven't bothered to address here at this stage because we haven't quite figured out how to do experiments to get at that problem. But there is this experimental need now that we do have to address. This is absolutely right.
Yeah. So I was wondering, I don't know if this is correct, but like in the model, the oscillation, the whole population enters this oscillation, right? And in the real case, it's only a part of the population that oscillates. So I was wondering, like this oscillation of acetyl-CoA, like the minimal threshold is because due to the cells that stay quiescent. Like if you divide, for example, the minimal acetyl-CoA by 35 and the maximum acetyl-CoA by 100, it will be the same.
That's right.
Level.
Right.
So a problem will be to answer why 35%, right, of the cell.
Even in the model, you can get 35%. It depends on where this lower branch is.
Right.
So if the low branch is high enough, maybe I've just drawn it. But if your branch was close to 100% here and it was close to 60% here, the oscillations you would observe would still be 160. And there would be lots of, so only 40% of the cells would actually grow. But in the model, because it's a well-mixed thing and everything, there's no distinction between different cells. It's just two things we're keeping track of, number of cells, so there are Q and number G. It's not, in the model, every cell at some point is probably going to switch to the other cell. If I was making actually a sort of agent-based version of this model, every cell would be allowed to switch and every cell would be switching at some point or the other. Whether there is in the real system a certain population that never switches is an experimental question that would be worth asking, and that would make the model different.
So that would actually be really useful to build a better model, right? And you've actually hit a really important question. We don't know if there are only two states in the system or a permanent third state which never divides or a small fraction that's always dividing somehow or the other. This model is so coarse-grained that it doesn't distinguish between these possibilities. And you've hit this spot-on problem that we have to tackle as experimentalists because our data is all bulk data. We have acetyl-CoA data from the population, gene expression data from the population, metabolite data, everything from the population. So we have to think of ways now to mark cells or trace cells across oscillations, but the challenge here is it's a well-mixed system and millions of cells. So how do we follow cells or groups of cells as they go through each cycle, right? That is a problem we have to get to address, yeah. Right, right, so that is true. So that we can control, and that actually the model can kind of predict without too much difficulty. You can change 30% to 15% or 40%, yeah? So the thing is though, the breakdown happens, so the window is not very large, both from the model and from experimentation. Once we increase glucose above a certain point, it breaks down completely. And this is not a very large window. Actually, even the model says that some 30% change in glucose, there's a low glucose and just a 30% or something increase, I don't remember the number, somewhere there, breaks it down. And both sides of this oscillation. And so this window is relatively small, which means we then start getting very, very messy biological data, which we can't make any sense of. Every cell is kind of doing its own thing out of sync. And we can't separate or sort, yeah? Yeah, you can get metabolic oscillations without that, but that's also, you might have metabolic oscillations at a much lower threshold. So there's studies which show that you can get metabolic oscillations without the cell cycle switch, but then you're working at a much lower threshold of oscillation. Something is going on, yeah. So I would love to have that. For an experimentalist, what you're saying is what we want, right? I've been trying to get Inca and others to think about how we can do that. We've got nothing. We've got nothing. Yeah.
When it goes from quiescent cell to, say, dividing or growth, so the energy will definitely define. So acetyl-CoA or TCA cycle, that takes over, and then it actually goes down. It comes to, when enough acetyl-CoA is made, you need to make NADPH because pentose phosphate pathway which will give ribose as well as
that's right.
NADPH will give fatty acid synthesis or lipid synthesis. So all biomolecular synthesis will need NADPH. Right. So I think this oscillating between glycolysis and pentose phosphate pathway in two different stages, which actually very obvious that it will regulate the oscillation between this.
Right. So this is true, and so intuitively this is correct. The point is to show how you can get a two-state switch with a threshold of one metabolite, which can hierarchically set up this entire cascade that comes afterwards. Also controlling the gene expression events, which we see. And in this case, this can actually control it because we have experimental data on the kind of acetylation events happening at histones, which are very correlated to the genes which are expressed at those times. So we can see that based on how much acetyl-CoA is there, which regions of the chromosome are being activated for gene expression, positional information, and the classes of genes represented over there, which is really, really striking. That's what couples both
gene expression and
yeah. We have both those pieces of data. That's bulk data, but that data is there.
We can stop here. Thank you, sir, for the talk.